So today I will talk about the potential effects of the diet on the Vipera ammodytes. So we know that the venoms are highly complex and that they are also highly variable. So sources of these variations are both in distinct evolutionary histories of divergent lineages and natural selection that plays on the acquisition of specific molecules and toxins. When we think about snakes, we need to think what they are using the venom for. So most of the time, almost exclusively, they are using it for the subduing, so the overpowering of prey before the prey can attack back or escape from the range where the predator can find it. So we can say that the main driving force of the venom variation is diet or the physiology of prey. So we said that the different species have different venoms, but when we look at the level of one species, we can also find some differences in the venom composition. So we should also consider geographic origin, meaning that different population of the same species can have different prey availabilities, and this prey availability can also affect venom. Also, different sexes, males and females, sometimes have different prey preferences, as well as throughout the ontogeny, so the lifespan of one individual, venom composition can change. However, the work by Mike and colleagues from last year showed that studying the 89 already published papers on venom composition of different different species from the subfamily Viperinae, most of the works somehow are not addressing this or not knowing this information. So sex and ontogeny are most of the time not pointed out. And in terms of origin, the situation is a bit better. So we would know the country and sometimes even the more precise locality of the venom from the specimens from which the venom is extracted. So knowing this, I was interested in studying exactly the venom composition based on geographic origin, different sex, and throughout the life of one individual. And the model species that I chose is the Vipera ammodytes. This is the widely distributed species, and it is presented with 4 subspecies. Also, within Balkan Peninsula, there is a considerable genetic diversities, as you can see in this map. So there are 7 distinct genetic clades. And also, important thing is that there is a sexual and ontogenetic differences in diet. So as for the venom, we know, as said, it is widely distributed. There are many snake bites in Europe, sometimes even with a lethal outcome. And the ones delivered to the prey, or in this situation, the person, the human, it can cause hemorrhage, tissue damage, and neurotoxicity. So knowing all this, it is not strange that this venom is already being studied, I would say, extensively. And here you can see some three profiles of the already published venoms. However, here we are stumbling on one more problem when wanting to compare the venom. And this is the table that from, again, Mike's work. And here I would just draw your attention to the different, so we have 5 protons here, 3 subspecies. However, the analysis used are totally different. And also in terms of quantification approach, they are different. Therefore, we could not really compare and say what are the differences between these venoms. So with this, objectives of my work is actually and will be to do the initial screening of venom profiles by sex, age, and different populations. And here I would just give you a small disclaimer. Please don't be harsh, because in the following slides I will use this word preliminary multiple times. These are still ongoing analysis, and there will be still some additional data analysis in order to get the full protons. But to show you now the where and what we did. So our study area, where we collected the venom samples of the Vipera ammodytes montadoni subspecies, is found in North Macedonia. And it comprises of 2 populations. So we have a Golemgrad island, which is population which is a locality where vipers are preying almost exclusively on ectotherms. And in the near proximity, some 3 kilometers away, there is a Koinsko Koinsko village, where we also have a population of nose-horned viper, and here these animals are also including small mammals in their diet. Therefore, also both populations are in high densities, and therefore it was a good system, good locality to study the effect of diet on the nose-horned viper venom. The method that we use is the bottom-up protocol, which allowed us to identify and do the relative quantification of all the venom components. And then, so how actually, what venoms we analyzed so far. As you can see, the venoms were pooled into 5 groups according to sex, age, and population. And here I will just add that after this field season, so after the summer of 2022, we added 2 more groups. So now we have also juveniles and subadults from the mainland. And right now I'm doing the analysis with Mike on these venoms. So let's go on the results. Here you can see the chromatogram of the male venom. So the pool of venom samples from adult males. And when comparing them, while overlapping with the females chromatogram, we can see no major differences in terms of peak number and intensities. Therefore, we decided to do the pools of the future groups, pools of males and females in the other groups that we were studying. So when we look at the age differences, comparing the juveniles with the the next size category, subadults, we can see that there are differences and that the venom is getting more complex. So new additional peaks are appearing, the intensities are different, therefore giving us a first impression that as the individual grows older, there is a need for more complex venom, which is also shown in the next group. But here it's not so much, so when we compare subadults and adults, not so much in terms of different peaks, but mostly in terms of intensities, meaning that subadults are exactly that, therefore this transition group. So just to make it more clear, when we compare the 2 most extreme size groups, juveniles and adults, we can see with these bars all the differences that we found that are present in the venom of this population. So here goes the preliminary part. As you can see, these differences are also shown in the protons. However, you can you can see straight away that still some part of the protons are under review. But however, we can say that with the increase in body size, there is the decrease in snake venom serine proteases and L-amino acid oxidases. So further analysis will be done, but this is something that we will point out for now. When we go to the population differences and when we compared island and mainland, this is something that really surprised me. So I was expecting much more differences between these 2 populations, knowing that they have different diet. However, most of the venom profile, well, most of the differences found in these profiles are based in the intensity of peaks. And this is also more or less seen in the protons, again, in the preliminary result of preliminary analysis of these protons. So if we want to, in terms of discussion, at the local scale, so we found no sex-related differences. We are hypothesizing that most probably similar diet in adults leads to also having similar venom profiles. Age-related differences, maybe, yes, there are some. There is some mild decrease in certain protein families, and this could be explained by a mild dietary shift throughout ontogeny. So yes, they are preying on ectotherms, but there is a slight change in terms of proportion of which kind of ectotherms. So there are centipedes and lizards, and juveniles are equally or preying on centipedes much more than the adult individuals. So this could be the reason for the change in the throughout the ontogeny. And then the most puzzling and most, for me, strange one is the population related. One, since we found only mild changes in some protein families, as I said, having different prey availabilities, I was expecting that the profiles will be different, but maybe there is just a mild differences in prey consumption. So at this moment, or maybe these populations are not separated long enough for the natural selection to act on this phenotype, or maybe there is some other thing, some other process that is going on and that is maybe diluting the effect of natural selection. We don't know at this moment. So far we have additional samples that we are analyzing, and then all of this will hopefully give us more clearer picture on what is going on here. And additional thing that I would just, so not to close it with this, but to close it with an open question and additional future studies. Since we had this opportunity to do that and to use the same protocol for analysis, bottom-up protocol, and the same quantification approach, we could compare, as seen on the top, this pie chart from the population that we studied, with the already published work by Benjamin Hempen and Mike from the same subspecies, just different geographic locations. And this opens a question, what is the driving force of these differences? Because we can see that in terms of, for example, phospholipase A2, there are considerable differences, as well as the vascular endothelial growth factor in the island venom is only 2%, while in the population from Turkey it's almost 11% present in this pie chart, in this venom composition. So this is something that will be interesting to see in the future. So we will try to collect the venom samples from population throughout the range of these species, and then we will hopefully see what is going on there and how different or similar they are. And with this, I will end my presentation. I would like to thank you all for your attention, and especially to thank to all my friends and colleagues, of course, my collaborators and my friends and colleagues who helped me in the field to collect venom samples. And special thanks to Mike for all the help and support during the lab analysis. So, thank you very much.
Okay, thank you very much, Maggie. Are there any questions from the audience on the chat? Nothing is until now, but maybe some of the people in the audience want to address the question to her. Yeah, Eric, just talk to the microphone.
Yeah, thank you for the great talk. I have a question regarding the kind of like your proposal of using different localities later on in your study. Are you considering maybe, I mean, they're not subspecies anymore, but the former subspecies like Rufoi in Italy and Gregovanneri in Austria, do you also maybe have a chance to analyze those? Because I think that might be very interesting to see if these like habitat types are differing in venoms from the normal Embodites.
Yes, well, thank you for the question. The thing with the taxonomy of VenomWednesday is a bit complicated, and there are many proposed subspecies. Even now, it's maybe going to the species level. So definitely, independently of how we call them, I would like to collect the venom samples from the Austria and Italy, and then also correlating it with the habitat type, with the prey, the small mammals that are available, and then seeing what is the composition of these venoms. So the problem with this, yes, actually knowing which subspecies is something that I would leave for the people who are more working with the genetics. I'm not that much into it. I will definitely also collect the tissue samples, so some analysis will be done, but this is a bit of a walking on a thin ice in terms of VenomWednesday, I would say, especially coming from a place where people are really in love studying these species. So yes, using and covering as much as possible of the different habitat types is priority.
Okay, thank you very much.
You're welcome. Thank you for the question.
So another question from the chat from Chris Weiser. How do you
Sorry,
know
I did.
about the different prey usages between the Golem Island and the mainland? Where does it come from?
Sorry, now the last thing I did not hear. Where did it come from, or
Yeah. Where does it come from?
the different prey usages?
Between
So
the
in
island
terms,
and the mainland?
yes, the island is has this specific situation that there are no small mammals found there. So the vipers are more or less doomed, not doomed, but they need to prey on what they can find and catch. Most of the times those are centipedes, which is also a bit problematic because centipedes are venomous themselves. And we also found a friend of mine found some years ago a viper killed by a centipede that it preyed on. So the centipede ate it from the inside. So they are preying on centipedes, and also there is Podarcis muralis, the common wall lizard, and this is the majority of their diet. So therefore, there is also the consequences of this kind of restricted prey types, since that these vipers are actually dwarf vipers, and they are around 20% smaller than any other population of Viperamodytes studied. And then, so this is just the situation like this, that there is no, for some reason, there are no small mammals, only mammalian prey that they can find. And they found 2 or 3 times in their diet are the babies of rabbits, but really neonates of rabbits. So because they're super small, and then the small vipers can prey on small babies of rabbits, but there are no any other source of mammalian prey there. And then we have this nearby locality of Koinsko. This is, I would say, the normal situation. In most of the localities, you can find diverse type of small mammals, and then vipers are opportunistic. They would try to prey on anything, not anything, but they would, they would try to include as much as possible small mammals in their diet, since they are highly energetically valuable, and so this is the situation in most of the mainland populations. I don't know the reason why there are no small mammals on island.